Hello, my name is Victoria Santos, and I am going to present to you the work titled Too Hot to Handle, Determining Devitrification Thresholds and Warming Rates of Reproductive Cells. I would like to thank the organizers for inviting me to talk to you about our work regarding the storage and handling of vitrified materials. I want to tell you that I am a chemical engineer and my PhD was also in engineering. So I want to invite everybody to think like an engineer when handling vitrified samples. I have developed several numerical models to simulate processes in food engineering, which I later applied to the reproductive area in order to determine cooling and warming rates of cryobiological systems. In the field of cryobiology, I have also studied the cryopreservation of seeds, and now I am in a preliminary stage doing some research in cryopreservation of recalcitrant yeast that are of biotechnological importance, and the cryopreservation of rainbow trout semen of a specific strain that has thermally adapted to higher temperatures. Also, Marina Sancinena, which is a co-author, she has achieved the vitrification of equine oocytes that have been successful, since you can see in the slides her 2 6-month-old foals, and the oocytes were named Tango and Milonga. I work in the National Scientific and Technical Research Council in Argentina, and all the authors are currently living in Argentina. It's at the tip of the Southern Hemisphere, and it's a country with a great diversity since we have ice glaciers, and up north we have the Iguazu Falls, which is the largest waterfall system. We are a very heterogeneous group. Dr. Sariski and I, we are both chemical engineers. Dr. Chirifi is a food scientist, and Dr. Marina Sancinena, she is our specialist in the reproductive area. Okay, so we have no conflict of interest in all our research articles. Okay, so everybody knows what happens to samples when they are frozen slowly, and when they are frozen at high cooling rates. So I'm just going to dive into the vitrification and tell you something about it. The vitrification is important to cryobiology because the natural state of liquid water inside living things is disordered. Therefore, when we vitrify, we maintain this natural disorder as the sample solidifies. How do we do this? Well, we have a supercooled solution that when it's cooled very quickly, the viscosity may reach values that are very, very high. So if we do it quickly, we do not allow the molecular rearrangement into a crystal structure. Therefore, we avoid the ice crystallization. It is a second order phase transition since there is no latent heat involved. The glass transition point, the TG, is the temperature where there is a reversible transition from an amorphous solid or glass into a viscous or rubbery state as the temperature increases. Below TG, the materials are rigid and brittle and amorphous. So what can happen to a vitrified sample during warming? Well, it can suffer devitrification. This is a phenomenon where there is ice formed during the warming stage. And also we have a recrystallization, which is the conversion of many small ice crystals into larger ones. These two phenomena occur at different temperatures and at different rates. The ice nucleation rate, for example, is high near the TG of the sample, and the ice growth rate is high just below the melting temperature. So, what are the minimum rates? The lower the concentration of the cryoprotectant, the faster you need to cool in order to avoid ice formation. And the minimum cooling rate necessary to avoid the significant ice formation is the critical cooling rate. The minimum warming rate in order to avoid the vitrification of a vitreous sample is called the critical warming rate. The critical warming rates are typically 2 or more orders of magnitude greater than the critical cooling rates. So if you have successfully vitrified your cell, it can devitrify during warming to form small intracellular crystals above Tg. And as the temperature increases, then these small ice crystals can grow in size by recrystallization, and that could be lethal. Fast warming is critical. This is a point often overlooked during the cryopreservation of reproductive cells. As you can see in this figure, the critical warming rate are higher than the critical cooling rate. They are 2 or more orders of magnitude greater than the cooling rates in order to avoid the vitrification and survival of cells. This was also published and said by Peter Masur and Seki in this research paper, where they stated that this is a new paradigm for the cryopreservation by vitrification. Because before, everybody was focused on cooling very fast, getting this vitrified sample, but now you have to focus on the warming because, as you can see, it's more critical. When can warming occur in a vitrified sample? Well, it can warm while we check before and after a transport from clinic to clinic, while we are doing witness step in IVF, and another embryologist has to check the ID of the sample, while we are transporting vitrified samples in dry shippers. For example, there is no liquid nitrogen allowed in airplanes. So these are steps where it's too hot to handle slowly. You have to be efficient, you have to move fast so you don't expose your straw, your sample, or your cryo device to high temperatures. And you have to be careful by monitoring the entire thermal history during the cold chain. So, since penguins are always present in a presentation in vitrification, I had to put mine in. But I have to say, we have to move with a rocket speed when we leave the liquid nitrogen to warm very fast, because warming is critical. So Marina Sancinena told us when we were in our group, we have to focus on the warming. What happens to the samples when they are handled in the storage in dewars? And it's very difficult to measure the temperature in cryo devices because we have very minimal volumes and they are very small in size. It's very difficult to put a thermocouple and measure time, temperature versus time. And you can observe a sample while crystals are forming during cooling or during recrystallization and fractures using cryostereoscope. But you can't do that every time you are going to vitrify a sample. So that's impractical and you are prone to high experimental errors. So what are the alternatives? Well, we can predict time temperature curves by numerical modeling of the heat transfer in these cryo devices. And we did that in our group, but we did it for cooling rates first. So we found the performance of each cryo device for cryotip, cryotop, cryoloop, etc. And we found the cooling rate and the performance for each of these devices. After 2013, we said, let's use these numerical models and try to see the warming of these cryo devices. But therefore, we applied the numerical methods to cryotop and to OPS. And that's where we're going to focus. Our objective was to find the glass transition temperature of a vitrification solution and to describe the devitrification thresholds and how they affect the storage and handling conditions of vitrified samples. How did we do this? Well, we first measured the TG using differential scanning calorimetry. And we used mathematical modeling to predict the time temperature curves and the devitrification threshold during warming of OPS. This was done using the numerical method called the finite element technique. And we did it in a software called COMSOL Multiphysics. So we solved the partial differential equation to describe the heat conduction process of OPS when we lifted the OPS from the liquid nitrogen and maintained it in a nitrogen vapor phase. We also measured the temperature inside a cryogenic storage dewar, and when we disturbed the nitrogen vapor by raising and lowering the canister. Here we have a diagram of the heat flow versus temperature and the key thermal transitions during the vitrification of a sample, or of a vitrification solution, I mean. We use a differential scanning calorimeter, and we can see here that the step change is the glass transition. The onset of the vitrification is TD, and it has an exothermic peak. And then we have the onset of melting with the endothermic process. Here we have the actual curve thermogram for our vitrification solution. You can see we have the glass at -125 approximately, and the devitrification peak and the melting or diffusion. And we could calculate the heat of the vitrification and the latent heat of melting. Well, here are the heat transfer equations for the OPS device. I am very sorry for everybody, but I am going to show you the equations, the governing partial differential heat transfer equations for this bidimensional axial symmetric domain, because it took me about 5 years of PhD to understand, solve, and find and achieve the prediction of the temperature inside these devices by solving all these equations. They are very complex. We need a lot of time, and well, it took me 5 years that I can resume here in one slide. Subscript F and P are the biological and the plastic support, and we have to know that there are thermophysical properties involved in these equations, which are the density, the specific heat, and the thermal conductivity that must be known and used as input information in the numerical model. If we solve all these equations that are second-order derivatives and in transient state, we can find the temperature as a function of radial and axial coordinates and time at each point inside the device. So the OPS system, we have 2 volumes, 1 microliter and 2 microliters, and we calculated the length over the diameter, and it was 2.4 for 1 microliter and 4.9 for 2 microliters. So this cannot be approximated as an infinite cylinder. We had to use a model where it was a finite cylinder. And an important aspect of the equations is that we need the boundary equation. The boundary equation involves the use of the surface heat transfer coefficient, which is a very important engineer parameter. It is a characteristic parameter of the convective heat transfer between the fluid medium and the surface of the object that is being cooled or warmed by this fluid, which surrounds the entire object. The H appears as a proportionality factor in the equation. Here you can see H times the temperature of the surface minus the external temperature. The temperature of the surface is the surface of the object, the OPS or the cryotop, and the external temperature would be the temperature of the nitrogen vapors. And that, as you know, we have liquid nitrogen at -196, and as we move in a vertical direction, we have an increasing temperature gradient of nitrogen vapors. What we have in a Dewar tank is a stratification phenomenon of the nitrogen vapors. The top of the Dewar represents the hot surface, and the liquid nitrogen would be the cold source. So when you have a hot surface facing down, you have a stratification phenomenon because the density decreases with increasing temperature. So the less dense layers are above the more denser ones, and therefore there is no convection, and the heat is transported by conduction only. This fact means that the H are decrease in value in comparison to the opposite, for example, for a heated pot where the cold source is facing up. So what do we need in the numerical model? We need the heat transfer coefficients that we have determined in our group, and we also corroborated these values with literature values using the Nusselt correlations. And we selected and calculate they were between 10 and 15 watt over square meter degree Kelvin. We selected two different nitrogen vapor temperatures, -70 and -50 degrees, and they were considered for the calculation of the critical time needed for the system to reach a final temperature that would be -100, -120, or -130. These are values close to the TG of the biological fluid. The initial condition, well, it was submerged in the liquid nitrogen. It was -196 degrees at times at T equals 0 seconds. And we have the thermal properties of the plastic and the thermal properties of the glassy water. All of these properties are involved in the governing heat conduction equations. Here you can see the temperature at different heights measured from the top of the Dewar. For a full Dewar, you can see that at 14 centimeters, you have reached temperatures above TG. And for a half full Dewar, there is also almost 2 centimeters away that reach -125. That would be around the TG of our vitrification solution. So above, we have risky zones that can enter, the sample can enter a rubbery state. What happens if we disturb all this nitrogen vapor in a half full Dewar? Well, we reach the TG at about 26 centimeters. That would be point B in the Dewar, below the neck. This is another way of showing these results, that for a full or half full, we have the dangerous zone here at the neck between 14 and 16 centimeters. High risk of the vitrification since the temperature here of the nitrogen vapors are above TG. And for a half full after raising, we have a larger zone that goes to about 26 centimeters. So we know that the OPS, when it's placed over a certain height above the liquid nitrogen, it begins warming. And as the height increases, the temperature of the nitrogen vapor also increases. So if the OPS reaches the TG, the vitreous biological sample may enter into a rubbery state, it transitions into the state, and it would then have the ability to form ice through devitrification. How do we quantify this time? How much time do we have? Well, we can quantify this critical time using numerical modeling of the warming process, using, as I said, the software console with the finite element method. And here you have the figure where we have predicted the temperature versus time in the OPS at a critical point, which is, and it tells us that -120 is reached at about 54 seconds for an H equals to 10 watt over square meter degree Kelvin, and a nitrogen vapor temperature of -50. We made several predictions for different temperatures and two different H values. And these are the times in seconds of these critical times that are very important to know when we lift samples. So the results tell us there is a temperature distribution of nitrogen vapor inside a dewar that is non-uniform, and it's because we have a stratification phenomenon of these nitrogen vapor layers. These temperature gradients are similar in a full and half-full dewar. However, when we disturb the nitrogen vapor, we establish a new actual distribution of temperatures, and we have higher temperatures overall. The nitrogen vapor temperature established after these disturbance is a critical value because we increase the risk of damage to vitrified samples. The mathematical modeling tells us that an OPS could reach a devitrification temperature in about 30 seconds when exposed to -100 to -130 degrees Celsius. Well, we also measured the TG, and it was in the range of -126 through to -121, after which, if we increase, we enter rubbery state and then devitrify. In this medium, cells should be stored at temperatures below -121 degrees Celsius or lower. And because storage under rubbery conditions is unstable, samples should be handled and stored below their TG. Now we wanted to test the survival of bovine oocytes during handling in dewars, and we applied 3 treatments. A one-step removal from liquid nitrogen. Treatment 2, a one-step removal from nitrogen vapor. And treatment 3, it was a 2-step removal with a 15-second pause at the dewar neck. We can see here the effect of the 2-step removal from a vapor nitrogen storage. The temperature here was above the TG, and the hatch at day 9 gave a survival of 0. So the other 2 treatments, which were samples exposed to temperatures below TG, there were no significant differences, and there was survival. Final results. There were no difference in development vitrified worn oocytes after one-step removal conditions, either with samples submerged or stored in nitrogen vapor. These results agreed with the recommendations previously proposed with the numerical models that the samples should remain below TG. In contrast, with 2-step removal condition from nitrogen vapor phase, the temperature of the sample surpassed the TG and the survival was reduced. Under these conditions, the samples were exposed to temperatures shown to be in the range of the rubbery and devitrification thresholds. These results, therefore, reinforce the importance of exercising extreme cautions while handling vitrified material during routine laboratory procedures, such as identification and confirmation and inventory keeping. Final remarks. Storage of vitrified samples in liquid nitrogen vapor phase constitutes a potential risk since devitrification can occur if we disturb these nitrogen vapor layers. Devitrification thresholds should not be interpreted as a fixed value since it is influenced by the TG of the media and the heat transfer efficiency of the vitrification support, because it's not the same if we have a cryotop or we have an OPS or a cryoleaf or any other vitrification device that has different loaded volume in it or different thickness in the plastic supports. The mathematical modeling can be used to analyze complex thermal phenomena. It can be applied to evaluate the critical warming times, new storage designs, and inventory procedures prior to exposing real-life samples. Well, thank you all for your attention, and here we have the references.